please join me in welcoming to the Distinctive Voices podium, Dr. Susan Weiss.
Thanks, Jennifer. For me, it's always great to be in California. It's kind of my second home. Okay, so my title is 40 Years with Coronaviruses, and I will start here. So here's an overview of the lecture. I'm going to give a brief introduction to coronaviruses, then a sort of history of human coronaviruses from the 1960s until the current SARS-CoV-2 pandemic. So they've been around for a long time. And then a comparison of the three epidemics that caused by spillover of zoonotic coronaviruses into humans, and then a little bit on determining the origin of SARS-CoV-2, which has been controversial, and then how coronavirus biology informs us on designing antiviral strategies, and then at the end, just a little bit about SARS-CoV-2 variants and a little bit of work from my own lab. Okay, so coronaviruses they look like this. This is sort of an iconic picture from an electron microscope. And those little spiky things sticking out from the virus particle give it the sort of name corona because it looks like a crown. And they're a family of viruses within a larger group of viruses called nidoviruses. And nido means nested. And this is because they express a nested set of RNAs during infection, but I'm not going to really talk about that. And this is a schematic of the virus particle. So coronaviruses have an RNA genome. That means there's no DNA at all in their life cycle, only RNA, which has consequences I'll mention a little bit later. And it's a very large RNA, and it's complex with a protein called nucleocapsid here, and it comes into this sort of helical capsid inside of the virus particle, and then around the virus particle in the blue is a membrane that's derived from the host cell when the virus is actually produced. And then in this membrane, there are three proteins, the spike protein that I think everybody's heard about by now, and that's the protein that attaches to the cellular receptor when the virus enters the cell. And it's important then for viral entry, but also from spreading from cell to cell, and I'll show a picture of that later. And it's also the main target for most of the vaccines, for the mRNA vaccines and also the adenovirus-based vaccines. And that's because it's sort of, it induces antibodies that will neutralize or inactivate the virus. And then there's other proteins here called membrane and envelope protein, are two other smaller proteins in the viral membrane. And some coronaviruses have another protein called hemagglutinin esterase that forms those yellow smaller spikes that SARS-CoV-2 doesn't have this protein, but some other coronaviruses do. And it's similar to a protein expressed by influenza. Okay, I'm going to give a coronavirus timeline, and I thought for this part I'm also going to talk a little bit about my own timeline, what was going on in my life and my research during the same time that we were studying coronaviruses, starting from the 60s and 70s. I wasn't doing them yet then, but if we look in the literature, we can see papers about OC43 and 229E, two common cold coronaviruses, way back at that time. But my first exposure or knowledge of coronaviruses came when I was a postdoc at UCSF in the late 70s, and I was working with Mike Bishop and Harold Varmus on RNA tumor viruses, and it was time for me to look for a job. So I didn't really want to work on tumor viruses anymore, partially because it was really competitive. It was before HIV emerged, actually, or just around the time. So I was looking for something different to work on, and I wanted to work on an infectious disease. So I looked in the literature and I saw something about coronaviruses. So there were these human viruses, but there was even more literature on animal coronaviruses. So coronaviruses infect just about every species of animal, and they're important pathogens for cats, dogs, pigs, cows. And also there was a mouse virus that was studied by some people. And it looked like a really interesting system because it had very interesting and novel mechanisms of replication, plus it had very interesting animal models to study hepatitis and even a model for multiple sclerosis. So Mike Bishop was generous enough to let me spend my last year as a postdoc working on this new field all on my own. So that was really amazing. And then in 1980, I was hired at Penn by Neil Nathanson, who also hired me to work on this really obscure group of viruses because he was really interested in pathogenesis. So that, in retrospect, I feel really lucky that he actually hired me. And so I was hired as an assistant professor that year. And another notable thing that happened that year was the first International Coronavirus Symposium, which was held in Würzburg, Germany. It was only about 60 people at that meeting, and that was the entire field at the time. And then, so from that time on till the first SARS epidemic in around 2002, there was a lot of basic virus biology done by a relatively small group of people that we were pretty much ignored. And most of this work was done with the mouse hepatitis virus, the model coronavirus, IBV of chickens, BCV of cows, TGV of pigs, very little on the human viruses at the time because they're cold viruses and nobody really cared very much about them. But during that time as well, I was promoted to associate professor and full professor. I had my first son and my second son. And then in 2002, the first SARS emerged in southern China. And it was really very shocking to the field. We were shocked because up till that point, we only knew that coronaviruses in humans caused the common cold, even though they caused some much more severe diseases in other species. But this was really new. And the same year, there was the 10th International Coronavirus Symposium in the Netherlands, which had hundreds and hundreds of people attending because we just had this epidemic. And that was the beginning of what I would call the pathogenic human coronavirus period. And SARS caused severe acute respiratory disease. And in the wake of the SARS epidemic, we learned that bats carried coronaviruses and many other viruses as well. And people started looking for other human coronaviruses, and only two were identified. HKU1 and NL63 were also considered more upper respiratory, although they did sometimes cause more severe diseases, but generally not very severe and not lethal, so different from SARS. And then in my life, I became the postdoc associate dean. Jennifer gave my whole history, but here it is again. And then things were sort of quiet until 2012 when MERS, Middle East Respiratory Syndrome virus, emerged in Saudi Arabia. Again, another lethal human coronavirus. This caused a lethal lower respiratory disease in humans. I'm going to talk a little bit about the epidemics in a minute. And then and MERS also came from bats. And then I was appointed vice chair of the micro department sometime after that. And then 2019, SARS-CoV-2, which really shocked the world. It was obviously we're still kind of living in the end of it, but why were we surprised? We'd already had it happen twice, but we were still surprised. Coronavirologists as well as other people were very surprised, and I'll talk about that in a little more detail, but it caused what we call COVID-19 or coronavirus disease 2019. And just a little plug, I think it's really important to differentiate between COVID and SARS-CoV-2. It's just like AIDS and HIV. One's a virus and one's a disease. Okay, so then we started a new center for research on coronaviruses during the pandemic. And then my granddaughter was born in 2022, which was super exciting, which is probably the best thing that happened during the pandemic for me. And then there was also the 16th International Coronavirus Meeting in Switzerland in 2023. And finally, I was elected to the National Academy, which is how I ended up here. So that's sort of a history of me and a history of the coronaviruses, and I'm not going to talk about myself anymore, but the rest is going to be just on the viruses. So, okay, coronavirus genome structure. I'm going to try to explain it as carefully as I can, as non-technically as I can. So there are different genera and subgenera of coronaviruses. To 2290 and NL63, the cold viruses are called alpha coronaviruses. The rest of them are beta coronaviruses, and they're in three different subgenera, OC43 and HKU1, the two SARS viruses, and MERS, all in different subgenera. But the important thing here is that all these viruses have very similar structure. This is a schematic of their RNA genomes. And all of them encode 16 or produce 16 proteins from this part of the genome. These are non-structural proteins, meaning that they're not actually in the virus particle, but they're really important for replication of the virus. And they're all pretty similar among all the different coronaviruses, and that'll become important later. And then the structural proteins, the ones I talked about, spike, E, M, and N, are all encoded in the same order in this part of the genome. So all these viruses are pretty similar. What makes them different is these numbered genes here. And these are genes that are important for the virus to combat the host immune system, innate immune system. So these are different for each type of virus, which gives them each a kind of different character and probably is important for how pathogenic they are. This is something that my lab studies a lot, but I'm not going to talk about it too much, just to say that this is what differentiates the different types of, the different genera, subgenera of coronaviruses. Okay, so coronaviruses are zoonotic. That means they cross host species infection. I'm going to give a couple of examples. So if we take a sort of ancestral beta coronavirus 1, it's called, that comes originally from a rodent. These are not from bats, but from rodents. And that virus infected both pigs, cows, and humans. And the virus can actually flip back and forth between humans and cows. And then this bovine virus has infected all these different species. And so each time it infects a new species, it kind of evolves to be a little bit different in each species. But the point here is that these viruses are very easily changed species, and they also can recombine, which means they can exchange genetic material with another virus and create new viruses. And it's believed that the bovine virus and the human virus kind of diverged in the late 1890s. So now we have this cold virus. It's closely related to this bovine virus, also very closely related to the mouse hepatitis virus. And then another example, even more complicated, we take an alpha coronavirus ancestral virus that came originally from a bat. This virus can also infect pigs, cats, and dogs. And these viruses, again, exchange really readily between the species. So that recently there's been a report of a virus that's partially canine, feline, and porcine all together here that can actually infect humans. So jumping species is not specific or peculiar to SARS, to the lethal viruses, but they all do it. This virus here, there's another virus called porcine epidemic diarrhea virus that's not directly related to this original virus. So a pig here can get infected with that virus, and then there can be recombinant viruses that come out of them. And just another example is kind of recent, interesting. This virus called HKU2, which came from a bat, infected pigs, and to cause a virus called SADS, or severe acute diarrhea syndrome coronavirus, and that virus has been known to infect humans. So there's a lot of exchange here between animals and humans, and just about all of them many of them come from bats and some from rodents. So with that as background, I want to talk a little bit about each of the lethal epidemics. So SARS-CoV first had its origin in a horseshoe bat. It was transmitted to a civet cat it's called, and the civet transmitted it to a human. And then it was transmitted to many humans by close contact. This is the 2002 version. And then it mostly infected people in Asia and Hong Kong, China and Hong Kong. And it was all over in 8 months, so it was really fast. And there was about 8,000 infections and about 10% mortality. And the reason it was ended so fast is thought to be because people were sick. They weren't spreading it pre-symptomatically, pre-symptomatically. So people were sick, they're in the hospital, they're isolated, and the virus was pretty much contained just by public health methods. And just a little more detail on that. So what it's thought that happened is that in a market in southern China, Guangdong province, there were the animals, the palm civet and raccoon dog, that were somehow infected with SARS from a bat, most likely before they came to the market, and then once in the market, spread to a human. It says animal handler, it may not have been an animal handler, but somebody was infected, and that person, somebody who was infected early on in China, went to a hotel in Hong Kong called Hotel M. This is sort of a a story that's spoken about a lot. And in Hotel M, this person infected a whole lot of other people on the same floor, and they just went to all different places and seeded little infections. But as I said, most of the SARS, the first SARS, occurred in China and Hong Kong, and there was an outbreak in Toronto as well by somebody who transmitted it here. Most of the people that got SARS in Toronto were either relatives or healthcare workers working with infected patients. So, so SARS was found in civets and raccoon dogs, and SARS-like viruses were found in bats, which were considered a reservoir for the virus. The civet was just like a quick transmittal. It's not like civets all over China had SARS. But many bats do have many coronaviruses. So let's move on to MERS. So MERS, interspecies transmission occurred in 2012, as I said, and MERS had its origin in a different kind of bat, Neuramesia capensis. And MERS is really interesting. It has a reservoir in camels, so lots of camels have been infected with MERS coronavirus, and it can transmit for camels. It does transmit to camels. It causes an upper respiratory little cold in camels. They're not very sick, but when transmitted to humans, it has a pretty high, like 35% lethality. It causes a kind of severe pneumonia like SARS. There's limited spread human to human. Most of the spread for this virus goes from camels to humans, mostly in the Arabian Peninsula. There was an outbreak in Korea, again by somebody moving from the site of the spillover from Arabian Peninsula to Korea and causing a little mini epidemic in Korea. And camels are a reservoir for MERS, and I'll just talk about that a little bit more. And there are still new cases in 2024. So MERS has never been eradicated. It continues to be have a reservoir in camels, and continues to very slowly cause infections. Their total has been 2,600 since 2012. So not a whole lot, but very lethal. So usually the more lethal, the less transmissible to humans, human to human, and about 36% mortality. A little bit more about MERS. So the way we know that there's so much MERS in camels is by measuring antibodies, that they have antibodies to MERS. They've been exposed to it. And this is an early study from just after the epidemic, where it was found that, this is by some people in Hong Kong, it was found that in some places, MERS, almost all the camels had antibodies to MERS, meaning they were exposed to it. For example, in the Middle East, in Jordan, in Saudi Arabia, but also in Africa, in Morocco, Egypt, Nigeria, Sudan. But then camels in Mongolia, Kazakhstan, Australia are completely negative for the virus. So parts of the world, there's lots and lots of MERS in the camels, and parts of the world not so much. Here's just another more numbers that were studied. But here's what's really interesting, I think. So MERS coronavirus strains in Africa and Middle East fall into 3 clades. Clades are just genetically related strains from a common ancestor. So there are different groups of related MERS. So that's like clade 1.1, clade 1.1, et cetera. And again, MERS came from bats. And interestingly, in the Middle East, MERS can transmit from camels to humans, like I just talked about in the original epidemic. But in Africa, the humans don't seem to get infected by the virus. So the viruses in Africa are a little bit different from the ones in Saudi Arabia, not very different, but different enough. And so that's one of the things that we're starting to work with in our lab, trying to understand why these are so transmissible to humans and these are not. So the clade A and B viruses found in the Middle East infect camels and can spill over into humans, whereas the clade C viruses infect camels. But because of this situation, MERS remains a potential pandemic threat. The World Health Organization has said that. And the reasons are that MERS could evolve and adapt to cause more human-to-human transmission. The African clade C MERS could adapt and or recombine with other viruses to produce strains that are able to infect and cause disease in humans. and camels or camelids, which are related to camels, naive to MERS, like in other parts of the world, could possibly get infected, leading to human infection. So MERS seems sort of quiescent now, but it's sort of, I think it could be a potential pandemic threat because it's so common in camels in so many parts, in both Africa and the Middle East. So, okay, so now we come to SARS-CoV-2. So SARS-CoV-2 has an ancestor, we believe, in a bat similar to SARS-CoV-1, the horseshoe bat. It's transmitted through an intermediate species again. Initially, it was thought to be a pangolin, which I think is not true, and I'll talk about that a little bit more. It's more likely through a raccoon dog, and then into humans, and then, of course, it spread all over the world. This is a highly spreadable, transmissible virus. Just to quote the numbers, there have been 7 million deaths from 700 million infections worldwide as of this year. And in the US alone, 103 million infections and 1.2 million deaths. So really, we all lived through this. We all know how horrible it was or is. So one of the big questions was, like, where did SARS-CoV-2 come from? And we don't really know the exact answer yet, but we know some things. We know what genera or what lineage is it. So it's a sarbecovirus, as I showed you earlier. It has this structure and these accessory proteins, very similar to the first SARS-CoV. What is its parent virus? And is there an intermediate species? So unlike SARS, the first SARS, the animal, so it's suspected or believed, and I'll talk about in a little more detail, that SARS-CoV-2 also came from animals in a wet market, but in Wuhan, China, not in southern China. But during the first SARS epidemic, the animals could be tested, and they were shown to be positive for the virus, for the SARS-CoV. But this time, the animals in the market were all destroyed, which made it impossible to really test them for live virus. So that's why, that became this huge controversy to figure out where the virus actually came from. So this is some of the work that was done early on to figure it out. So the SARS-CoV-2 first arose in humans here in Hubei province in Wuhan, China. So one of the viruses that was thought to be perhaps a precursor was something called RATG13, which came from Yunnan, which is over here, not so close, because it had very high homology by the RNA genome to SARS-CoV-2. Then there were viruses isolated from pangolins in Guangdong, which is where the first SARS came from, that also had some homology, not quite as much. So there was a whole lot of talk about where did this virus come from. That's where Guangdong is down in the south. So I'm just going to go through a little bit of the reasoning to try to figure out where did SARS come from. So I want to look a little bit at the coronavirus spike protein again, and here's a picture of it. It's got this sort of stalk structure here, which is attached to the membrane, the viral membrane, so that the protein sticks out from the membrane, like I showed in the first schematic. And this subunit here is this is called the S2 subunit, is responsible for the actual entry of the virus. So when the virus enters the cell, the viral membrane, shown here, fuses together with the cell membrane, and the virus gets pushed in. And I'll talk about it a little bit more. And at the top here is what's called the receptor binding domain, or the receptor binding motif. In the S1 subunit, this sort of globular structure, and this thing actually attaches to the receptor, which is called ACE2. You've probably heard of that. It's angiotensin converting enzyme. It's the receptor for SARS-CoV-2, and for SARS-CoV, but not for the other coronaviruses. So each coronavirus has its own receptor on the host cell. So we want to look at the spike protein a little more carefully to try to figure out where SARS-CoV-2 came from. So here's a linear picture of the spike protein, and there's these two subunits, this S1 and the S2. The S1 with the receptor binding area, and the S2 is the part that actually produces the functions in entry, viral entry. And some coronaviruses have what's called an S1, S2 recognition site here between S1 and S2 subunits. And in order for this protein to be functional, it has to be cut or cleaved at this site, at this S1, S2 boundary site, and S2 prime, a second site. And then you'll see that this is all really actually in that three-dimensional structure that I just showed you. But this cleavage has to happen, and the two subunits are separated, but they still say it's kind of associated together. But the reason I'm talking about the furin site is this was unusual for a SARS-CoV virus. Some coronaviruses have this site and this cleavage, and some don't. And people thought that it was very suspicious that SARS-CoV-2 had this furin site. So the RATG13 spike protein at no furin site, it looked very homologous, very similar to SARS-CoV-2, but the receptor binding domain was different. It didn't have the exact amino acids or the exact residues that would bind to the receptor. So it wasn't that close, even though people thought it might be a precursor. And the same thing for this pangolin virus, no furin cleavage site, and it didn't quite match in the receptor binding domain. So neither one of these really fit the bill for immediate precursor. So then people looked further, and some bat viruses were identified in Laos that have a very similar receptor binding domain to SARS-CoV-2, and a virus called BANL52 had an identical residues binding to the ACE2 receptor. Without going into all the details, just that this viral protein, this viral spike had identical amino acids that are pointed out with these arrowheads to SARS-CoV-2, so it looked like it could be a precursor, but we still don't really know. It's probably not the exact precursor, but it's like we're getting closer to maybe finding a precursor in bats. And just to talk a little bit more about I don't want to get too technical, but the S1, S2 furin recognition site became very controversial because, like I said, it was unusual for a SARS-CoV virus to have one. But if we look at all coronaviruses, this furin site is really just a group of basic amino acids. It doesn't really matter what they are, but designated by R, K, and H. So if we look at the different kinds of coronaviruses, the HKU1 and OC43, the so-called Mbeko viruses, have very basic S1, S2 cleavage sites shown here. And MERS coronavirus has maybe not as good a one, but some basic amino acids, and SARS-CoV-2 also. SARS-1 doesn't have this site. The alpha coronaviruses don't have the site. But to me, it's not very suspicious because if we look at the murine coronaviruses, just 3 different strains, these two have cleavage sites. This one does not. So very closely, viruses may or may not have these sites. The reason I'm talking about this is there was a lot of publicity about how this furin site told some people that there was something suspicious about this coronavirus, that maybe it wasn't natural. But it's perfectly natural for these viruses to have these cleavage sites or not. Okay, so one more thing about the origins. A paper came out recently, just in September, that was looking more carefully at whether this virus came out of the market in Wuhan. And so before this paper was published, we knew that SARS could infect civets and raccoon dogs and those kind of animals. We knew that the early cases of SARS were very much localized around this particular market in Wuhan. And within the market, there was a hotspot for virus, and that there were 2 different separate or related lineages of viruses found in the market. But this new paper said that they found that they found raccoon dog DNA closely associated with hotspots for viral RNA in the market. So wildlife, it was actually mitochondrial DNA, but it doesn't matter. Wildlife DNA was identified in samples from stalls that were positive for SARS-2, and DNA from raccoon, dogs, civets, and other wildlife species were detected in market samples. So this was just another step to say, yes, there were certainly viruses associated closely with animals in the market, but they could never actually look at the live animals to prove it. So this doesn't actually prove that the virus came out of the animals in the market, but it's some, I think, important evidence. So just to summarize, I feel strongly about this. How do we know SARS-2 was not engineered by humans and very unlikely to come from a lab? SARS-CoV-2 had never been seen in a lab before and doesn't resemble any other known natural or recombinant coronavirus. It's just not close enough to have been something that was slightly manipulated in a lab. And really, nobody knows how to design a virus with properties of SARS-CoV-2. We just don't know. If we look at the sequences of it, we don't know why it behaves the way it does. And without a template, without a virus, we can't construct a new virus. And similar viruses are found in bats. And as I said, many other natural coronaviruses have spike proteins with furin sites. And viruses evolve by natural selection, not by human design. And again, the epidemiological data suggests that virus spilled into humans in the seafood market in Wuhan because the coronavirus sequences collected from the market are in very close proximity to DNA from these wild animals. So just one other thing on that topic, I just wanted to show how to construct a virus. Like people say, oh, they just made a virus in the lab, but it's really not easy to do that. Because here's, say, a viral RNA genome here that I showed before, and you can't manipulate RNA. You can't cut and paste RNA. You have to only can cut and paste DNA. So you'd have to copy this RNA into this very long DNA, which is not so hard to do nowadays. And then you would have to mutate the DNA, which you could, but again, you might make a mutation here, but you couldn't really mutate it all over the DNA, and you couldn't ever really know how to mutate it to make it act like SARS. And then you would take this DNA and have to put it into some kind of cells, and then a virus would come out. So you could make viruses. It's not that difficult to make recombinant viruses, but you have to know what you're making to start with. So for all those reasons, I think that this virus is clearly from a natural virus from animals that probably spilled over in that market. Okay, so I'm going to switch now and talk a little bit about the biology of coronaviruses and why it's important to study it and what we can learn from it. Because it helps us design antiviral therapies, which we need. And I'm going to talk about two different aspects, viral entry and coronavirus conserved replication proteins, those 16 proteins that are encoded at the end of the virus. And then I think also understanding the biology tells us how we get SARS-CoV-2 variants, which will be the last part of the talk. So this is probably the most technically difficult to explain, but coronaviruses use two routes of entry into the cell. So one is called the plasma membrane route, and by this route, the virus here, the spike protein, attaches to the ACE2 receptor on the surface of a cell, and then the spike protein is cleaved by a protease, a protein that cuts proteins, and it cleaves the virus at the S2 site. And then the spike protein, when it's activated like that, mediates a merger of the viral and host membranes, and then the nucleocapsid, the RNA, is released into the cell, and it's ready to initiate an infection. That's called the plasma membrane route. The other route is by endosomal route, and again, it starts with the spike protein attaching to its receptor, and this time it's kind of sucked into the cell, into like a vesicle called an endosome here, and then the spike protein, again, mediates a fusion of the host and viral membranes, which has to happen at a low pH by an enzyme called cathepsin, and then, again, the RNA is released into the cell. So by either route, we get the same kind of infection. So what determines, I'll tell you in a minute why that's important, but first, what determines which route? And that has to do with the proteases, the TIMPers that's shown here, cathepsin that's in the endosome, and that can be different in different cell types and for different viruses, but also the furin enzyme that I talked about before that cuts that S1, S2 boundary to activate the spike, and that protein is found inside the cell. So viruses that have that S1, S2 cleavage site are actually pre-activated while they're produced. So when the virus comes out, it has to be clipped once more by TIMPers. And so this depends on the sequence of the spike. And back to these mouse proteins I mentioned before, these mouse viruses, this JHM strain has a really good cleavage site. It's cleaved very well by furin, and so JHM enters the cell by the plasma membrane route. This other virus, related, very closely related, called MHV2, its spike has no cleavage site, and it enters through the endosomal route. And then a third related virus called A59 has a partial cleavage, and it enters by both routes. So this is important for a couple reasons. One is that, SARS has this S1, S2 site, which is not great. Probably it uses both routes. It at least uses the plasma membrane route. It may use this one as well. It may use it in different cells to different degrees. But at least we know that in lung cells, this is a more efficient route of infection for SARS-CoV-2. But certainly the first SARS, which doesn't have a cleavage site, is perfectly good at causing lung disease. So this is kind of really a mixture. But the reason that it's important at all is that it would tell us what kind of inhibitors might work, or antivirals, because people have you can use cambistat, which is an inhibitor of this protease, TIMPRS, to prevent entry by this route, or you could use chloroquine or cathepsin inhibitor to prevent infection by this route. So it makes it a little bit complicated because you don't really know which route the virus is going to enter by, probably both. So for me, it seems like this is not a great approach. Entry inhibitors are not a great approach for coronaviruses, and in fact, they really haven't been used very much. But on the other hand, I'll talk about the inhibitors or the antivirals that have been used for coronaviruses. So as I said before, the left end of the genome encodes these 16 proteins, and they're conserved. This is really important among all coronaviruses. They're very, very similar, and they're good targets for antiviral therapies because they're so conserved, and they're enzymes. And the reason that coronaviruses need, or RNA viruses in general, need so many proteins for replication is that the host cell is not prepared to replicate RNA, so it has to bring in all its own stuff, so to speak, so it can replicate. Whereas DNA viruses, the other kind of viruses, can kind of use or hijack, as they say, host enzymes for replication. But anyway, so coronaviruses encode these 16 proteins, and these arrows denote that there have to be, again, cleavages. So viruses are very involved with proteases and cleavages. And in this case, the proteases are viral proteases, not host ones. So we know, for example, that this, what's called NSP3, non-structural protein 3, encodes a papain-like protease that cuts these very large proteins here and here. And then what's called a 3C-like or main protease cuts at the rest of these arrows. And then the RNA-dependent RNA polymerase, or the enzyme that actually replicates the genome and transcribes the messenger RNAs, is encoded in NSP12. So these are the proteins that have been used so far as targets for antivirals. You've probably heard of remdesivir, that was originally an Ebola drug, and molnupiravir, which is a relatively recent Merck drug, that are both directed against the RNA-dependent RNA polymerase and have been used for SARS-CoV-2. And then I think everyone's heard of Paxlovid or Paxlovid from Pfizer, and it's directed against this protease here. So these have been successful ways to target coronaviruses, antivirals, and I guess this is modeled on proteases and polymerases have been the targets that have been used successfully against HIV and also against hepatitis C. And so just to summarize, the proteases process the replicase proteins here, and the DRP replicates the RNA genome and RNA. But there are all these other non-structural proteins, and many of those are enzymes as well. And just to show them here, without going into detail, some of these proteins are parts of the replication machinery, like the primase and the activator, helicase. And then others of these proteins are really important for preventing the host cell innate immune system from preventing replication. So they're so-called antagonists of the host cell response. And many of these are enzymes, and they are many of them are potential targets for antiviral strategies. And they really, I think people are starting to look at these as targets, but they haven't been used nearly as much as the proteases and the polymerases that I just mentioned. So again, to summarize, there are enzymes that promote synthesis and also the stability of the viral RNAs as they're being replicated, and also protection from the host cell sensors or the host cell from sensing that it's infected and responding with interferon and other antiviral factors. So yeah, so these are all, I think, potential targets that will be used at some point. And the really important thing, again, is that since these proteins are very similar for all the coronaviruses we know, and probably for future ones, they're really going to be used, again, sort of, they call them pan-coronavirus inhibitors. They'll be around when the next coronavirus comes by. We'll be more ready than we were this time. For real. Okay. And then, and just to point out, these proteins are all together in a big complex, so-called replication complex. It's pretty. That's a cryo-EM picture. Okay, so the last part of the talk is about the SARS-CoV-2 variants. How do they arise, and why are some of concern, as they called, you've heard the expression variants of concern. Okay, so here's the viral RNA, again, just shown in blue, so-called genome RNA. And here's the spike protein in green, just to show where it is. So basically, when the virus starts to replicate, the genome RNA produces an RNA-dependent RNA polymerase that I just mentioned, and that polymerase or enzyme copies the RNA from this side to this side into a copy, which is called an antigenome. It's just if it were DNA, there'd be two strands, so that you get a sort of an antigenome, or the opposite strand of RNA shown here. And then that polymerase, and in doing so, it causes double-stranded RNAs, which are something that the host cell sees as a danger signal, just as an aside. And that's how the host knows it's infected and starts getting its interferon system and other antiviral systems activated. But anyway, so this is just a natural part of replication. It makes this double-stranded RNA. It then copies back and makes more RNAs. And I should mention, too, that these RNA replication enzymes they're highly error-prone, and that's part of the way they work. And I think the reason they make errors is that so the virus is able to evolve and change. But anyway, in doing so, each new genome is going to have like errors or mistakes or putting in the wrong nucleotide. And when those errors are kind of either negative, the RNA will not survive. If they're neutral, it probably won't survive either. But if it happens to be, say, in the spike protein, and it gives that particular virus an advantage for replication, then it'll take over the whole system, and it'll eventually be a variant, right? So a variant occurs when there's a mutation, and then there's some kind of selection for spread or some other thing, which I'll talk about in a moment. So that's really, so making variants is a totally normal part of the virus life cycle. They're constantly changing, and then with selections, the strongest survival of the fittest kind of virus survives. Oh, and the other thing that's a consequence of this mechanism is that it allows recombination to occur. So as this polymerase is copying to minus strand or back the other way, if it can jump to a new RNA, and that's the way you get the joining of sequences from two different genomes. And that's how we get recombinance. It's another way that we get changes in the genome or variants, and then those can be selected for evolution as well. Okay, so this is just a picture of my chairman of my department, Rick Bushman, was sequencing RNAs like crazy, and this just shows this is in the Delaware Valley outside of Philadelphia where we live. Just to show, initially we had the Alpha, Delta, and Omicron variants. We're still in the Omicron variant system, and they keep evolving and changing, and I don't actually know how it's decided when it's still Omicron or when it's different enough to be the next variant. But right now we're still in different Omicron systems. and I guess in general, it's believed that the Delta variant was more pathogenic, causing more lower respiratory disease, while the Omicron variant was more upper respiratory, less pathogenic, but spread even better than the earlier viruses. And it's hard to know in a way because when a virus becomes sort of less pathogenic, at the same time we were getting vaccinated, so it's hard to know how much of a virus, how much it's due to vaccine and how much is due to the virus actually changing. But I think in the case of Omicron, it really is more upper respiratory, at least from studies in animal models and things like that. But in any case, they're pretty different, those two variants. And I just want to talk a little bit about what makes them of concern. So here's the spike protein again, and most of the focus in the variants is on the spike protein, where most of the evolution happens. So here's alpha, beta, and delta, and Omicron variants, and they have mutations in really similar places. These are all in that receptor binding domain that I talked about before, and these here are in the that S1, S2 furin cleavage site. They make the cleavage site more basic from a proline to histone. It doesn't really matter, but this makes a better or more cleavable spike protein. And these here make a protein that can either bind to the receptor better or can evade the antibody response from the vaccine better. Okay, so why are they of concern? Because just sort of what I just said, increased receptor binding, this one particular amino acid substitution or mutation allows the spike protein to bind better to the receptor, which might make it more spreadable or more spreadable. Or immune escape, which means that it will escape the vaccine or the antibodies that you get either from infection or from vaccine. And then the increased S1, S2 cleavage by this cleavage site becoming more basic, would it make entry better or also cell-to-cell spread fusion, which I'll show in a minute. So those are kind of the three different reasons, but also mutations in other genes could contribute as well, could make the virus spread better or make it evade innate immunity or anything like that. But most of the focus has been on the spike and the ability to evade antibodies. So this is some data actually from my own lab. And this is all done with a collaborator, Noam Cohen. He's an ENT surgeon, and he has a huge biobank of cells from the noses of his patients. These are primary nasal epithelial cells that we then culture on an air-liquid interface, so they look kind of like the nose, like the inside of the nose. And the first part is an electron microscope picture of these cultures. And they contain ciliated epithelial cells. Here's diagrammed up here. And then also cells, goblets, they're called goblet cells, and they produce mucus. So this really simulates the inside of the nasal cavity. And you can see here, these are the cilia beating. I think this is really neat. So they're actually beating in culture. And then in this culture, we can see the cilia are pushing around mucus at the top of the culture. So we've been using this for the last few years to infect with different coronaviruses. And so the ciliary beating and the mucus are the sort of the first defense against a pathogen that gets into your nose. And we use this to compare the different variants. So here, this is real data. This is showing the amount of virus, and this is in a logarithmic scale. So this is like 100, 10,000, a million, 100 million infectious units of virus, and this is times of infection, overinfection in those cultures I showed you. And we take the virus, we measure the amount of virus produced at each time point by different variants, wild type, alpha, beta, delta, Omicron. And what we found was that the Omicron variant replicates the best here in the red. These are just statistically significant when they have asterisks on them. So all the viruses replicate better than the original, what we call wild type, or the original parent virus, but Omicron beats out all the other viruses in replication. But then when we look at, we looked here as these are these same cultures infected with wild type, delta, and Omicron, and then the red, what we do is we stain these cultures with an antibody against a viral protein. So the red represents infection. The green represents cilia, And then the blue is just nuclei of the cells. And we can see that the cultures look really different for each virus. So here's the wild type, and each red here is really another infected cell. And the same here for Omicron. But with Delta, we see these big masses of cells, and the cilia are gone. So Delta is the most sort of cytopathic, like it really messes up the cells. It destroys, it deciliates. We don't know how. And it looks like these cells are fusing together. So remember I said that the spike protein, when it cleaved well, it can cause a lot of fusion between the cells. We think that's what's going on with Delta. So Delta is clearly the most destructive, maybe consistent with it being the most pathogenic. And then this is just the way we look at cleavage. I talked a lot about cleavage, but these are called polyacrylamide gels. And basically, they separate proteins, different sizes. And this top here, at full length, this is the spike protein before it's cleaved. And this S2 is the spike protein cleaved. So we can see that Delta has the most cleaved versus uncleaved spike protein. So it's cleaved really well, causes the most destructive infection. Omicron is less cleaved, and the same for all the other viruses. This is just the nucleocapsid protein, just kind of to show that there's infection there. And then just, so if we want to look at cell-to-cell fusion, which is very pretty to look at, we infect cell lines, not these primary nasal cells, but just a cell line shown here. And we can, again, stain these cells with, the green represents the viral proteins here. The blue represents the nuclei. And you can see here that, for example, Delta, you see this huge mass. That's like I don't know, 50 cells fused together by the virus. And you can see the blue is just all the nuclei in the middle, and the green is the virus infection. And you can see the same thing, the alpha virus causes some fusion, but really none for Omicron, beta, and very little for wild type. So Delta is the most destructive. It causes the cell-to-cell fusion. It has cleaved spike. So they're all a little bit different from each other. I think that was, yeah, so that's the last slide. I'll just summarize here. So coronaviruses often have an origin in bats, infect many species of animals, and spill over into humans, possibly causing epidemics or pandemics, so far 3 times in the 21st century. MERS coronavirus has a major reservoir in camels and may present a danger for further spillovers. SARS-CoV-2 has closely related strains in bats and most likely spilled over into humans from a raccoon dog or other animal in the wet market in Wuhan. Coronaviruses enter the cell by 2 pathways dependent on multiple host proteases, making entry difficult to target for antivirals. In addition to viral proteases and polymerase, additional coronavirus conserved enzymes present multiple targets for antivirals, potentially, and would work on any future coronaviruses should they arise. And SARS-CoV-2 variants arise during viral RNA replication. Omicron replicates most efficiently, while Delta induces the most cellular damage, perhaps consistent with the degree of disease, hypothetically. And just one more slide, and then I'm done. To prepare for future emergent viruses, I think vaccine development is really important. Monoclonal antibody treatment was useful against coronavirus, against SARS-CoV-2. I think Donald Trump was treated with it. And it's important to keep developing pan-coronavirus antivirals for future outbreaks using all those other possible targets as well as the polymerase and protease. And then I think this is important. The last 2 points: continue to identify and characterize coronaviruses and other viruses from bats and other species. I think it's really important to know what's out there and what could possibly infect humans. And then the last point is something dear to my own heart: to continue basic virology research, which is what I've been doing for 40 years. And I think it often gets the short shrift that there's a lot of money put into antivirals and vaccines, but not a whole lot of money given into the basic virology. And just a picture of my lab, who's done some of this work. That's Noam, the guy with the ENT guy with all the cells. And I'll stop here for any questions.